This is on chapter 21, the immune system, innate and adaptive body defenses, and specifically in this talk, we are talking about the innate defense system. So why does this matter? Understanding the signs of a healthy immune response will help you assess your patients. The immune system provides resistance to disease, and it's made up of two intrinsic systems: the innate or non-specific defense system, and the adaptive or specific defense system. The innate defense system constitutes the first and second lines of defense. The first line of defense are the external body membranes, skin, mucous membranes. Second line of defense are going to be the antimicrobial proteins, phagocytes, and other cells. The adaptive defense system is our third line of defense, and it's going to attack particular foreign substances, and it takes longer to react than the innate defense system. So when we talk about the immune system, we're talking about a functional system. So we're not going to be talking about an organ system. So we're not going to be talking about organs and tissues and stuff like that in this particular chapter, not in terms of the tissues that constitute the organs. We will talk a little bit about tissue. The innate and adaptive defenses are intertwined. Both of them release and recognize many of the same defensive molecules. The innate defenses do have specific pathways for certain substances, and the innate responses release proteins that alert cells of the adaptive systems to foreign molecules. So here are our two systems. We have the innate defenses, or the innate defense system, and the adaptive defense system. The innate defense system is going to constitute the first line of defense, which is going to be our skin barrier or surface barriers, skin, mucous membranes. And then our internal defenses are our second line of defense. And these are going to be phagocytes, natural killer cells, inflammation, antimicrobial proteins, and fever. And then I lost my cursor. Okay, there it is. The adaptive defenses include humoral immunity, which is carried out by B cells, and cellular immunity, which is carried out by T cells. So part one of this whole chapter is on innate defenses. The innate system uses first and or second line of defense to stop attacks by pathogens, and pathogens are disease-causing microorganisms. Our surface barriers are our skin and our mucous membranes and their secretions. And this is going to provide a physical barrier to most microorganisms. And then if you remember going back to AMP1, and we talked about the layers of skin of the epidermis, and as the cells are moving up toward the surface, they start to become filled with keratin, right? And so keratin is resistant to weak acids and bases, bacterial enzymes, and toxins. And then our mucous membranes provide similar mechanical barriers. Skin and mucous membranes produce protective chemicals that inhibit or destroy microorganisms. For example, the acidity of skin and some mucus secretions inhibit growth. This is called the acid mantle. Lacrimal fluid. So we find lysozyme in saliva, respiratory mucus, lacrimal fluid. And what lysosome does is it kills many microorganisms. We also have enzymes in our stomach that kill many microorganisms. Mucin is a sticky mucus that lines the digestive and respiratory tracts and traps microorganisms. And defensins are antimicrobial peptides that inhibit microbial growth. And then there's also other chemicals like lipids in sebum and dermacidin in sweat that are toxic to bacteria. The respiratory system also has modifications to help stop pathogens. So for example, the hairs in the nose are coated with mucus, and that helps to trap particles that are inhaled. And then the cilia of the upper respiratory tract sweep dust and bacteria-laden mucus toward the mouth. Now, if the surface barriers are breached by nicks or cuts, then the second line of defense is going to protect deeper tissues. And this is a table that is showing the the first and second line. Well, not the first and second. It's just the first line of defense, and showing the intact epidermis, intact mucous membranes, and what each of these do. Actually, this is a lot more information than you need to learn. Just make sure you know the stuff that's in the actual text in the previous slides. Then we have our second line of defense. And like I just said on 2 slides ago, in the event that we have a breach in our first line of defense, we have a breach in either our skin or our mucous membranes, then the second line of defense has to come in. And we have a number of players, right? We have phagocytes, natural killer cells, inflammatory response, antimicrobial proteins, and fever. Many second line of defense cells have these things called pattern recognition receptors that recognize and tightly bind to structures on microbes, which will disarm them before they do any harm. And one class, toll-like receptors, play a central role in triggering immune responses, and humans have 11 different toll-like receptors. Phagocytes are white blood cells that ingest and digest foreign invaders. Neutrophils are the most abundant phagocytes. This goes back to chapter 17. Remember, never let monkeys eat bananas. So neutrophils are the most abundant, and they become phagocytic on exposure to infectious material. Macrophages, macro means big, phage means eater. These guys are going to develop from monocytes, and they're the chief phagocytic cells, the most robust phagocytic cell. And then we have free macrophages and fixed macrophages, and their names kind of tell you what they do. So the free macrophages are free to wander through tissue spaces. For example, alveolar macrophages are found in the lungs. Then we have fixed macrophages, which are permanent residents of some organs, like stellate macrophages found in the liver, and microglial cells that are found in the brain. This right here is a macrophage, this blue guy, and the little green rod-shaped guys are bacteria that are about to be engulfed. So phagocytosis is one of the questions that could be a potential essay question, just so you know. The process starts when the phagocyte recognizes and adheres to the pathogen's carbohydrate signature. Now, some microorganisms have this thing called an external capsule that will hide that surface carbohydrate, and that's going to help them evade phagocytosis. Then we have this term opsonization, and this is when the immune system uses antibodies or complement proteins as opsonins that coat the pathogen. It actually acts almost like handles for the phagocytes to kind of grab onto, which enhances phagocytosis. So this is step 1. Phagocyte adheres to pathogen. Then we have 2, 3, 4, and 5. And 2 would be cytoplasmic extensions called pseudopods. They bind to and engulf the particle into a vesicle called a phagosome. Then this phagosome fuses with the lysosome, forming what's called a phagolysosome. And then the phagolysosome becomes acidified, and the lysosomal enzymes will digest the particles. Anything that's indigestible or residual waste is exocytosed from the phagocyte. And exocytosed means it's kind of put into like a little vesicle, and then, or not even in a vesicle, it's just released outside. Here, I'll show you a picture. It isn't a vesicle, but it's because it was already in one. So we have the phagocyte, again, adheres. Then we have the little pseudopods. They look like little cytoplasmic extensions. They're coming around, and they're going to form this vesicle, this phagosome, right? Then this lysosome comes in and binds to the phagosome and forms a phagolysosome. And then the lysosomal enzymes digest everything in here. and then it's like I said, it's already in the vesicle, and any waste or residual material is exocytosed out of the cell. I switched that slide quicker than I intended to, but that's okay. I was done talking. So some pathogens are not killed by that acidified lysosomal enzyme, kind of being added to the phagosome. And then we have helper T cells will trigger macrophages to produce respiratory burst, which is going to kill pathogens resistant to lysosomal enzymes by doing three things: releasing cell-killing free radicals, producing oxidizing chemicals such as hydrogen peroxide, increasing the pH and osmolarity of the phagolysosome. And then in neutrophils, this is separate, okay? So this is no longer talking about the helper T cells. The helper T cells do those three things. Now, in neutrophils, defensins can also help by piercing the membrane of the pathogen. Natural killer cells are non-phagocytic large granule lymphocytes that police blood and lymph. They kill cancer and virus-infected cells before the adaptive immune system is activated. And what they do is they attack cells that lack this cell surface receptor. So our cells in our body have cell receptors, and so the natural killer cell is looking for any cell that doesn't have that, because it assumes that if it doesn't have that it's foreign and it needs to be taken out. And it kills by inducing apoptosis in cancer cells and virus infected cells, and apoptosis is programmed cell death. They also secrete potent chemicals that enhance the inflammatory response. Inflammation is triggered whenever body tissues are injured. Injuries can be due to trauma, heat, irritating chemicals, or infections by microorganisms, and there are some benefits to inflammation. It prevents the spread of damaging agents. It disposes of cell debris and pathogens. It alerts the adaptive immune system, and it sets up the stage for repair. There are four cardinal signs of acute inflammation: redness, heat, and swelling, and pain. And sometimes there's a fifth one. If the person is unable to use an area or move an area, then impairment of function may also be a sign of acute inflammation. And then we have the stages of inflammation, and we're going to go over each one of these individually, but they are inflammatory chemical release, vasodilation and increased vascular permeability, and phagocyte mobilization. Inflammatory chemical release. So injured tissues or immune cells can release chemicals into the extracellular fluid. For example, mast cells release histamine, and this is a key inflammatory chemical. There's other inflammatory mediators besides histamine: kinins, prostaglandins, cytokines, and if pathogens are involved, complement. All of them are going to cause vasodilation of local arterioles, and vasodilation means that the lumen diameter is getting bigger, which means that more blood is going to come to the area. They all make the capillaries leaky, which means that more fluid is going to leave the capillaries and go into the interstitial spaces, and many attract phagocytes to the area. Vasodilation and increased vascular permeability. So again, vasodilation means more blood's coming to the area, and this can cause hyperemia, which means congestion with blood. And this is going to lead to the redness and heat that we talked about a couple of slides ago. The increased capillary permeability causes exudate to leak into the tissue. And exudate is fluid containing clotting factors and antibodies, and this is going to result in local swelling or edema. And the swelling can push on nerve endings, which can cause pain. Other things that can cause pain are toxins from bacteria, or released prostaglandins and kinins. There are some benefits of edema. The surge of fluid in the tissue sweeps foreign material into the lymphatic vessels for processing in the lymph nodes, and it delivers clotting proteins and complement to the area. The clotting factors can form that fibrin mesh, which can act as a scaffold for repair, but that mesh also isolates the injured area so invaders cannot spread. And then we have phagocyte mobilization, and this is another potential essay question. So make sure that you are, you know it. So neutrophils flood the area first, macrophages follow. Okay, so neutrophils are first at a party, macrophages are the late arrivers. If inflammation is due to pathogens, then complement will be activated, and then adaptive elements will arrive. So steps for phagocyte mobilization. The first is leukocytosis. And this is the release of neutrophils from bone marrow in response to leukocytosis inducing factors that are released from injured cells. We have marginization, and this is when endothelial cells of capillaries in that inflamed area project these things called cell adhesion molecules into the lumen of the vessel, and they grab onto the neutrophils that are passing by, cause them to slow down and roll along, clinging to the vessel wall. Next, we have diapedesis. The neutrophils will then flatten and squeeze between the endothelial cells, moving into interstitial spaces. And then finally, we have chemotaxis. So chemotaxis is when something is either drawn to or kind of pushed away from an area because of a chemical. So positive chemotaxis means that something is drawn to an area. Inflammatory chemicals act as chemotactic agents that promote positive chemotaxis of neutrophils toward an injured area. As attack continues, monocytes arrive later. They get there about 12 hours after leaving the bloodstream, or about 12 hours after they leave the bloodstream. It doesn't take them 12 hours to get there after they leave the bloodstream, but 12 hours after leaving the bloodstream, they are transformed into macrophages. The late arrivers then replace the dying neutrophils and remain for cleanup prior to repair. And that is what this is showing. And in the event that this is an essay question, this is totally good as an answer. So, leukocytosis, neutrophils are going to enter the blood from the bone marrow. Marginization, those cams are being projected into the lumen, which is causing these neutrophils to grab onto the wall and slowly roll. And then diapedesis, flattening and squeezing through endothelial cells. And then finally, chemotaxis is going toward the chemical signature. Here are the questions for this first part. All right, I hope you guys have a good day.